much. Good afternoon, everyone. Unfortunately, this is the last talk of the conference. I must say thank you for the organizers for allowing me to present today. Also because it's the last one and it's the most different one as Tina's presentation. The advantage of having the last talk is, in my case, beneficial because this way you will remind that plants were part of the conference. So basically that is what I'm gonna try to convince you that plants can respond to sound and vibrations, especially in the context of pollination. So our project started at the end of 2022. It's called Good Vibes. And we are using one particular plant, which is a snapdragon, to try to demonstrate this hypothesis. So I think I can convince you, or maybe you are already convinced, that plants are very cool. They are beautiful. They have so many structures, particularly flowering plants, angiosperms, possess a huge broad range of structures, colors, pigments, produce aromas, and they do this mostly to interact with pollinators. So in the case of animals, mostly insects, most of these flower structures attract in one way or another these insects. We know now that insects and plants, maybe you already know as well, is one of the best cases of coevolution. They started at the Cretaceous, and the angiosperms basically, angiosperms radiated thanks to this coevolution with insects almost 125 million years ago. And to date, the angiosperms, the flowering plants, are the most abundant plants in the planet. They more or less represent 90% of land plants, which is more or less 300,000 species. So these interactions are mainly based on visual and olfactory cues, so pigments or aromas that attract pollinators, and then the pollinator is able to disperse the pollen, basically. But recently, in the last 10 years, some evidences have been telling us that maybe plants can also participate in some other types of communication, particularly with vibrations and acoustics. So there are some examples here. You can see cases of close contact, short range, and whether they are airborne substrate vibrations or substrate-borne vibrations. There are different cases in which plants can interact with insects or other animals through vibration and sound. One case, a particularly interesting case, is the case of caterpillars or herbivores that there are some studies demonstrated that plants can listen to the chewing sounds of caterpillars and respond through stress hormones to produce compounds that are anti-herbivore. And this is just with the sound, not with the presence of the caterpillar. There are other cases such as buzz pollination, in which we can see that the pollinator embraces the anthers of a flower, vibrates in a high frequency and releases the pollen. And this is the pollen that then it will transfer to other flowers. So based on these evidences, we hypothesized that maybe pollinators were also interacting with the plants in a way of trying to improve the pollination of one particular species, and we believe that plants may respond to particular vibroacoustic signals of their most efficient pollinators and trigger different responses, for instance, sugar in the nectar or aromas or many other compounds, to improve the successful rate with these particular pollinators. So in this context, we started this project 3 years ago, and this is basically a group of 3 very interdisciplinary, multidisciplinary groups. It's led by the entomology group from Francesca Barbera and Lucy Casacci, sound engineering group by Sebastian Oberst in UTS in Sydney, and myself and my group for plant physiology. So the 3 groups are working together to tackle whether plants can respond to this. Our species of interest, our model, is snapdragon, Antirrhinum. for different reasons. One of them is it's the domesticated species, its genome has been sequenced. It's a model plant for flower-related processes. There's a vast collection of mutants, especially flower mutants that we can use to test in the interaction with pollinators, but most importantly because they are quite dispersed in Spain, so we don't have to move much. So what we have been doing, it's basically to try to understand which is the network of most frequent pollinators in the Iberian Peninsula, particularly close to Valencia. And so what we do is to record both audio and video of behaviors of these different pollinators or rovers. I will explain later on which are the rovers. And we also identified pollen load on these specimens to see how good they are in transferring pollen. The audio recordings, we can represent them as linear spectrograms in which we can have different representations of the frequencies on the time scale. So the beginning will be when it's hovering around the flower, then when it's landing, and then when it's going out of the plant. And we use these audio recordings once engineered for playback experiments. So we put the plants in these playback experiments and we sample tissue, flower tissue for different purposes. Transcriptomes, sugars, volatiles, and so on. So as you can see here, we sampled different locations in the Valencian community. This work was done mostly by PhD student Lorenzo Bianco, and basically during 2 consecutive seasons we tried to understand which was the frequency of different pollinators in these regions. As you can see here, the number of visits from spring to summer decreases quite, and actually I could understand because Valencia in summer is a living hell. So we basically conducted our analysis at the very springtime. We identified more than 1,000 plant-insect interactions. We identified 44 species. Most of them are bees, Hymenopteras. We used more or less 500 or 400 flowers for doing analysis of how many seeds were produced when 1, 2, or 3 visits of each particular insect were occurring. And we got around 200 both audio and video processed files. As you can see here, different specimens are found in different time points during these intervals of the different months, and some of them are quite abundant and frequent. Actually, the ones that are on top here are the ones that we have been selecting later on for our playback experiments. We did also pollinator performance analysis, so we trapped the flowers after 1, 2, or 3 visits of each one of these different the most frequent pollinators. We studied different parameters such as number of seeds, visitation rate, temporal presence, and we came up with metric to identify the best performance pollinators. Among these, we found Rhodantidium sticticum. This is a solitary red bee. As you can see here, it is more or less of the size of the flower, so it can easily enter. There are other bigger Hymenoptera, such as bumblebee, that are so big that cannot enter, but somehow still tries to. This one in particular poses its body over the lower lobule, and then the flower opens because of gravity of the pressure, and then they can enter. We also identify some rovers, which, for instance, this wasp is able to perform the lower part of the flower, rob the nectar without the need to enter, so it's not a real pollinator. Here I show you more or less what I was saying, that it hovers around the flowers, it lands, and then it's able to open it and enter. Let's see if I can let you hear the sound. So this would be the sound of the Rhodantidium. So it has a very particular features in frequency, in time, And this would be, for instance, one of the rovers. So you can see it's completely different, right? So we use these different audio recordings to try to stimulate plants and see whether they were able to discriminate between one type of playback or another, and if they were capable of responding to the ones that belong to the most frequent pollinators. So for this, we study different plant responses. We are interested both in early and late responses to the potential playback. Sorry, there we go. So here you can see some of them. We're interested in sugars within the nectar, but also in complete flower transcriptomic responses, aroma profiles, and pigments. We did a initial transcriptome analysis of the flower, of the complete flower, but now we are trying to move forward and doing single cell transcriptomics to see if there are specific organs or cells within the flower tissues that are responding more than the other. And we perform mostly multi-omic integration and multi-omic analysis. These are just examples of some experimental setups. We had performed experiments in very highly controlled conditions in phytotrons or semi-field conditions such as greenhouses. And we have stimulated plants in different time points with all these different type of conditions. So the pest pollinators, the rovers, ambient noise, and ping noise. And we have sampled at different time points. So these are many different experiments that we have been conducting during the last 2 years. On our first pilot experiment, we were very interested and excited because we already saw some changes. So the playback of Rhodantidium was able to increase the nectar volume and also the total amount of glucose, fructose, and sucrose, which are the most abundant sugars within the nectar. And this was both compared to the ambient noise or to the ping noise. We further a little bit more into this analysis. So we did a time course in playback, ping noise, and ambient noise or silence. And we see that basically in the playback condition, we have a significant increase of both glucose and fructose, which is not observed in the ping noise or in the silence conditions. I will not go very much into the detail of this, but we also did the genome sequencing of these species, so the Antirrhinum litigiosum. We see which is actually very similar in size to the domesticated genome. They are very similar, despite some duplications, some translocations, but they are very similar. And thanks to the genome sequencing, we were able to perform a first transcriptome analysis and identify some homolog genes to genes that have been characterized in model plant species that are nectar-specific sugar transporters that could have some differential expression. So we want to work with these genes a little bit more in detail later on. We have been also studying the volatiles that these plants produce in response to the sound. So we perform gas chromatography mass spectrometry, and this is just an example of what we can see at 3 hours after the playback. And we were very interested to see that not only we could find volatile organic compounds induced, but also repressed in the comparison with the silence treatment. Among the induced, we found xenal, which have been described as aromas that are involved in the attraction of pollinators. And we also interestingly found alpha-farnesin repressed, which is a sesquiterpene that has a very broad range of effect over pollinators. So we think that maybe this one is repressed because this way the plant can control the specific attraction of one particular species and not all the broad range of potential insects. We have other candidates that we want to try to identify later on. Some of them are quite interesting, some aldehydes, some alkenes, This one in particular, undecane, is repressed, and it's related to sex between insects. So this is interesting. I have no idea what this means, but it's quite interesting to see this. And finally, we are using these nectars and these aromas to try to do preferential experiments with the insects. So here you can see some examples of these particular insects like Xylocarpa violacea, Bombus terrestris of Rhodantidium, that are feeded and are associated to, for instance, in this case, to one particular color, and then we follow up whether the insect chooses one color or the other. We are doing this both with the nectar and with the volatiles. I will not go more in detail on the next part, but just to tell you that the sound engineering team are doing very cool stuff in this project. They are doing deep learning modeling of all the spectrograms that we have been obtaining from these recordings, adding information on the behavior of the insects, and with this we have been able to potentially identify the insect species just by the recordings, and we do this with high accuracy and precision, but I will not go in detail because I'm already out of time. And I will just pass this one, and with that I thank you very much for your attention.